Alright, so now we're currently in the Sackler Institute for Comparative Genomics. It is one of the newest parts of the museum. We have an incredible view of Manhattan, and if you look over here, you'll see one of our laboratories. There's multiple, and we'll be going inside to take a closer look. So yeah, these are a sample of Papadum Bonus. They're one of the protist species that we work with, so a protist would be a single-celled organism. These were collected by one of our friends in Japan, Aki. He was on a submersible mission to the Marianas Trench and discovered these about 10 years ago. Currently we're working on sequencing their genome to find out a little bit more about them. They're a really interesting species because they are single cells and they eat a lot of things extremely rapidly, and very little is known about them since they're only 10 years old. This is the lab that I work in for a lot of my preparatory work. So the work that I'm doing currently is working with the Papadum Bonus, which I just showed you, which again is a complex kind of single-cell organism. And what we're trying to do is take a bacterial cell, particularly a bacterial cell, which can undergo photosynthesis, right? The process that plants use to make food, but there are bacteria species that do it on their own. And the bacteria are a lot smaller than protists, so they're a lot smaller than Papadum Bonus. And the idea would be to have the larger, more complicated cell engulf the smaller bacterial cell, and instead of eating it, which Papadum Bonus does a lot of, it would make that bacterial cell basically its slave and work for it and create food for the larger cell. This actually did happen on Earth probably multiple times, 1.5 billion years ago. And we're trying to recreate it for the first time in the lab. So it's a step in the process of the evolution of life, particularly complicated life, and even more particularly the evolution of algae and plants. So this would be one of the most crucial points in the formation of all plant life on Earth. Okay, so this is where I do the primary part of my work. So right here is a early Earth apparatus, which we built over the summer. Basically, the way this works is that we would put a solution of salt water in here that mimics the exact concentrations of the ocean 1.5 billion years ago. And then what we would put inside of that container would be these two guys. So right here on my left-hand side is the cyanobacteria, or the bacteria that can produce photosynthesis. And you can tell that it's very green, very much like plant life. So that the green is from the chlorophyll, which allows them to actually make food from the sun. And then here we have the Papadum Bonus sample, which again is the large complex organism which would hopefully consume the green cyanobacteria and turn it into its slave. So we would put it into this apparatus, which with the early ocean formula. Now this apparatus is connected through this tubing to a nitrogen tank over here. And basically we would bubble the nitrogen into the early ocean formula, and what that would do is it would remove the oxygen from the ocean. So the ocean 1.5 billion years ago in the Proterozoic had very, very, very little oxygen, and we're trying to mimic that as much as possible. And the way that we get it down to a very precise amount is through this program here, which is something that I, along with colleagues, have created. It basically controls the amount of nitrogen bubbling through it at any time, so that we don't have to sit here and manually do it. We can leave it overnight, and we can leave it for a week or so and see how it progresses, see if the large microbes actually took in the cyanobacteria and turned them into photosynthetic organisms. Right here we have a microscope. This is the microscope room. This is where we would actually analyze the early ocean formula we were talking about, and particularly we would be looking for less bacteria. Remember, the photosynthetic bacteria, we would look for less of them in the solution, more of them actually overlapping with the larger Papadum Bonus. And if they're overlapping, but you can still see the green, then that would mean that they haven't been eaten by the Papadum Bonus, but instead they've been inserted into them. More analysis would have to be done to see if they're actually exchanging food. But this is the step to actually confirm if what we're trying to do would actually did yield results. What this means going forward is there's two ways to look at it. There's one there's the practical. So in terms of the pure knowledge, we'd have a much better understanding of how life on Earth actually evolved and the steps that it took to get here, which I think is an important goal, philosophical goal for a lot of science. And the practical aspect would be something like if you can successfully create this process in the lab, then you can take smaller bacterial cells, presumably, and have a mechanism to insert them into larger cells. So the Papadum Bonus, the larger, more advanced cell, is known as a eukaryotic cell. Humans also have eukaryotic cells. These are just large, sophisticated cells with a lot of things inside of them, as opposed to the bacterial cells. If you could somehow create a bacteria that works for you, like for example, diabetes patients, we use bacteria to manufacture insulin. So if you could use a bacteria to enter into a larger eukaryotic cell, which could be a human cell, to do something for you, there are potentials for medical applications and other things along those lines. So that would be the interest for